tools that the geneticists of today routinely employ. One such powerful cytological technique is karyotyping, a method in which geneticists can identify traits characterized by chromosomal abnormalities from a single cell. To observe an individual's karyotype, a geneticist first collects a person's cells, like white blood cells, from a blood sample or other tissue. In the laboratory, he or she stimulates the isolated cells to begin actively dividing. The geneticist then applies the chemical colchicine to cells to arrest condensed chromosomes in metaphase. The geneticist then induces swelling in the cells using a hypotonic solution so the chromosomes spread apart. Finally, the geneticist preserves the sample in a fixative and applies it to a slide. The geneticist then stains chromosomes with one of several dyes to better visualize each chromosome pair's distinct and reproducible banding patterns. Following staining, the geneticist views the chromosomes using bright field microscopy. A common stain choice is the Giemsa stain. Giemsa staining results in approximately 400 to 800 bands of tightly coiled DNA and condensed proteins, arranged along all 23 chromosome pairs. An experienced geneticist can identify each band. In addition to the banding patterns, geneticists further identify chromosomes on the basis of size and centromere location. To obtain the classic depiction of the karyotype in which homologous chromosome pairs align in numerical order from longest to shortest, the geneticist obtains a digital image, identifies each chromosome, and manually arranges the chromosomes into this pattern, figure 13.5. At its most basic, the karyogram may reveal genetic abnormalities in which an individual has too many or too few chromosomes per cell. Examples of this are Down syndrome, which one identifies by a third copy of chromosome 21, and Turner syndrome, which has the presence of only one X chromosome in women instead of the normal 2 characterizes. Geneticists can also identify large DNA deletions or insertions. For instance, geneticists can identify Jacobson syndrome, which involves distinctive facial features as well as heart and bleeding defects, by a deletion on chromosome 11. Finally, the karyotype can pinpoint translocations, which occur when a segment of genetic material breaks from one chromosome and reattaches to another. 13.2. Chromosomal basis of inherited disorders 335 chromosome or to a different part of the same chromosome. Translocations are implicated in certain cancers, including chronic myelogenous leukemia. During Mendel's lifetime, inheritance was an abstract concept that one could only infer by performing crosses and observing the traits that offspring expressed. By observing a karyogram, today's geneticists can actually visualize an individual's chromosomal composition to confirm or predict genetic abnormalities in offspring, even before birth. Chromosome number disorders. Of all of the chromosomal disorders, chromosome number abnormalities are the most obviously identifiable from a karyogram. Chromosome number disorders include duplicating or losing entire chromosomes, as well as changes in the number of complete sets of chromosomes. They are caused by non-disjunction, which occurs when homologous chromosome pairs or sister chromatids fail to separate during meiosis. Misaligned or incomplete synapsis, or a spindle apparatus dysfunction that facilitates chromosome migration, can cause non-disjunction. The risk of non-disjunction occurring increases with the parent's age. Non-disjunction can occur during either meiosis I or II, with differing results, figure 13.6. If homologous chromosomes fail to separate during meiosis I, the result is 2 gametes that lack that particular chromosome and 2 gametes with 2 chromosome copies. If sister chromatids fail to separate during meiosis II, the result is 1 gamete that lacks that chromosome, 2 normal gametes with 1 chromosome copy, and 1 gamete with 2 chromosome copies. Visual connection figure 13.6. Non-disjunction occurs when homologous chromosomes or sister chromatids fail to separate during meiosis, resulting in an abnormal chromosome number. Non-disjunction may occur during meiosis I or meiosis II. Which of the following statements about non-disjunction is true? A. Non-disjunction only results in gametes with N+1 or N1 chromosomes. 336 Chapter 13. Modern Understandings of Inheritance access for free at openstax.org. B. Non-disjunction occurring during meiosis II results in 50% normal gametes. C. Non-disjunction during meiosis I results in 50% normal gametes. D. Non-disjunction always results in 4 different kinds of gametes. Aneuploidy scientists call an individual with the appropriate number of chromosomes for their species euploid. In humans, euploidy corresponds to 22 pairs of autosomes and 1 pair of sex chromosomes. An individual with an error in chromosome number is described as aneuploid, a term that includes monosomy, losing one chromosome, or trisomy, gaining an extraneous chromosome. Monosomic human zygotes missing any one copy of an autosome invariably fail to develop to birth because they lack essential genes. This underscores the importance of gene dosage in humans. Most autosomal trisomies also fail to develop to birth. However, duplications of some smaller chromosomes, 13, 15, 18, 21, or 22, can result in offspring that survive for several weeks to many years. Trisomic individuals suffer from a different type of genetic imbalance, an excess in gene dose. Individuals with an extra chromosome may synthesize an abundance of the gene products, which that chromosome encodes. This extra dose, 150%, of specific genes can lead to a number of functional challenges and often precludes development. The most common trisomy among viable births is that of chromosome 21, which corresponds to Down syndrome. Short stature and stunted digits, facial distinctions that include a broad skull and large tongue, and significant developmental delays characterize individuals with this inherited disorder. We can correlate the incidence of Down syndrome with maternal age. Older women are more likely to become pregnant with fetuses carrying the trisomy 21 genotype, figure 13.7. Figure 13.7. The incidence of having a fetus with trisomy 21 increases dramatically with maternal age. Link to learning. Visualize adding a chromosome that leads to Down syndrome in this video simulation. http://openstax.org/l/down_syndrome. Polyploidy. We call an individual with more than the correct number of chromosome sets, 2 for diploid species, polyploid. For instance, fertilizing an abnormal diploid egg with a normal haploid sperm would yield a triploid zygote. Polyploid animals are extremely rare, with only a few examples among the flatworms, crustaceans, amphibians, fish, and lizards. Polyploid animals are sterile because meiosis cannot proceed normally and instead produces mostly aneuploid daughter cells that cannot yield viable zygotes. Rarely, polyploid animals can reproduce asexually by haplodiploidy, in which an unfertilized egg divides mitotically to produce offspring. In contrast, polyploidy is very common in the plant kingdom, and polyploid plants tend to be larger and more robust than euploids of their species, figure 13.8. 13.2. Chromosomal basis of inherited disorders 337. Figure 13.8. As with many polyploid plants, this triploid orange daylily, Hemerocallis fulva, is particularly large and robust, and grows flowers with triple the number of petals of its diploid counterparts. Credit: Steve Karg. Sex chromosome non-disjunction in humans. Humans display dramatic deleterious effects with autosomal trisomies and monosomies. Therefore, it may seem counterintuitive that human females and males can function normally, despite carrying different numbers of the X chromosome. Rather than a gain or loss of autosomes, variations in the number of sex chromosomes occur with relatively mild effects. In part, this happens because of the molecular process X inactivation. Early in development, when female mammalian embryos consist of just a few thousand cells, relative to trillions in the newborn, one X chromosome in each cell inactivates by tightly condensing into a quiescent, dormant, structure, or a bar body. The chance that an X chromosome, maternally or paternally derived, inactivates in each cell is random, but once this occurs, all cells derived from that one will have the same inactive X chromosome or bar body. By this process, females compensate for their double genetic dose of X chromosome. In so-called tortoiseshell cats, we observe embryonic X inactivation as color variegation, figure 13.9. Females that are heterozygous for an X-linked coat color gene will express one of two different coat colors over different regions of their body, corresponding to whichever X chromosome inactivates in that region's embryonic cell progenitor. Figure 13.9. In cats, the gene for coat color is located on the X chromosome. In female cats' embryonic development, one of the two X chromosomes randomly inactivates in each cell, resulting in a tortoiseshell pattern if the cat has two different alleles for coat color. Male cats, having only one X chromosome, never exhibit a tortoiseshell coat color. Credit: Michael Bodega. An individual carrying an abnormal number of X chromosomes will inactivate all but one X chromosome in each of her cells. However, even inactivated X chromosomes continue to express a few genes, and X chromosomes must reactivate for the proper maturation of female ovaries. As a result, X chromosomal abnormalities typically occur with mild mental and physical defects, as well as sterility. If the X chromosome is absent altogether, the individual will not develop in utero. 338 Chapter 13. Modern Understandings of Inheritance, access for free at openstax.org. Scientists have identified and characterized several errors in sex chromosome number. Individuals with 3 X chromosomes, XXX, are phenotypically female but express developmental delays and reduced fertility. The XXY genotype, corresponding to one type of Klinefelter syndrome, corresponds to phenotypically male individuals with small testes, enlarged breasts, and reduced body hair. More complex types of Klinefelter syndrome exist in which the individual has as many as 5 X chromosomes. In all types, every X chromosome except one undergoes inactivation to compensate for the excess genetic dosage. We see this as several bar bodies in each cell nucleus. Turner syndrome, characterized as an X0 genotype, i.e., only a single sex chromosome, corresponds to a phenotypically female individual with short stature, webbed skin in the neck region, hearing and cardiac impairments, and sterility. Duplications and deletions. In addition to losing or gaining an entire chromosome, a chromosomal segment may duplicate or lose itself. Duplications and deletions often produce offspring that survive but exhibit physical and mental abnormalities. Duplicated chromosomal segments may fuse to existing chromosomes or may be free in the nucleus. Cri du chat, from the French for cry of the cat, is a syndrome that occurs with nervous system abnormalities and identifiable physical features that result from a deletion of most 5p, the small arm of chromosome 5, figure 13.10. Infants with this genotype emit a characteristic high-pitched cry on which the disorder's name is based. Figure 13.10. This figure shows an individual with Cri du chat syndrome at 2, 4, 9, and 12 years of age. Credit: Paola Cerruti Mainardi. Chromosomal structural rearrangements cytologists have characterized numerous structural rearrangements in chromosomes, but chromosome inversions and translocations are the most common. We can identify both during meiosis by the adaptive pairing of rearranged chromosomes with their former homologs to maintain appropriate gene alignment. If the genes on two homologs are not oriented correctly, a recombination event could result in losing genes from one chromosome and gaining genes on the other. This would produce aneuploid gametes. 13.2. Chromosomal basis of inherited disorders. 339 Chromosome inversions. A chromosome inversion is the detachment, 180 degrees rotation, and reinsertion of part of a chromosome. Inversions may occur in nature as a result of mechanical shear, or from transposable elements action, special DNA sequences capable of facilitating rearranging chromosome segments with the help of enzymes that cut and paste DNA sequences. Unless they disrupt a gene sequence, inversions only change gene orientation and are likely to have more mild effects than aneuploid errors. However, altered gene orientation can result in functional changes because regulators of gene expression could move out of position with respect to their targets, causing aberrant levels of gene products. An inversion can be paracentric and include the centromere, or paracentric and occur outside the centromere, figure 13.11. A paracentric inversion that is asymmetric about the centromere can change the chromosome arm's relative lengths, making these inversions easily identifiable. Figure 13.11. Paracentric inversions include the centromere, and paracentric inversions do not. A paracentric inversion can change the chromosome arm's relative lengths. A paracentric inversion cannot. When one homologous chromosome undergoes an inversion but the other does not, the individual is an inversion heterozygote. To maintain point-for-point synapsis during meiosis, one homolog must form a loop, and the other homolog must mold around it. Although this topology can ensure that the genes correctly align, it also forces the homologs to stretch and can occur with imprecise synapsis regions, figure 13.12. Figure 13.12. When one chromosome undergoes an inversion but the other does not, one chromosome must form an inverted loop to retain point-for-point interaction during synapsis. This inversion pairing is essential to maintaining gene alignment during meiosis and to allow for recombination. 340 Chapter 13. Modern Understandings of Inheritance access for free at OpenStax.org. Evolution connection. The chromosome 18 inversion. Not all chromosome structural rearrangements produce non-viable, impaired, or infertile individuals. In rare instances, such a change can result in new species evolving. In fact, a paracentric inversion in chromosome 18 appears to have contributed to human evolution. This inversion is not present in our closest genetic relatives, the chimpanzees. Humans and chimpanzees differ cytogenetically by paracentric inversions on several chromosomes and by the fusion of two separate chromosomes in chimpanzees that correspond to chromosome 2 in humans. Scientists believe the paracentric chromosome 18 inversion occurred in early humans following their divergence from a common ancestor with chimpanzees approximately 5 million years ago. Researchers characterizing this inversion have suggested that approximately 19,000 nucleotide bases were duplicated on 18p, and the duplicated region inverted and reinserted on chromosome 18 of an ancestral human. A comparison of human and chimpanzee genes in the region of this inversion indicates that two genes, ROC1 and USP14, that are adjacent on chimpanzee chromosome 17, which corresponds to human chromosome 18, are more distantly positioned on human chromosome 18. This suggests that one of the inversion breakpoints occurred between these two genes. Interestingly, humans and chimpanzees express USP14 at distinct levels in specific cell types, including cortical cells and fibroblasts. Perhaps the chromosome 18 inversion in an ancestral human repositioned specific genes and reset their expression levels in a useful way. Because both ROC1 and USP14 encode cellular enzymes, a change in their expression could alter cellular function. We do not know how this inversion contributed to hominid evolution, but it appears to be a significant factor in the divergence of humans from other primates. 1. Translocations. A translocation occurs when a chromosome segment dissociates and reattaches to a different, non-homologous chromosome. Translocations can be benign or have devastating effects depending on how the positions of genes are altered with respect to regulatory sequences. Notably, specific translocations have occurred with several cancers and with schizophrenia. Reciprocal translocations result from exchanging chromosome segments between two non-homologous chromosomes such that there is no genetic information gain or loss. Figure 13.13. Figure 13.13. A reciprocal translocation occurs when a DNA segment transfers from one chromosome to another, non-homologous chromosome. Credit. Modification of work by National Human Genome Research, USA, 1 Violaine Goids et al. Segmental duplication associated with the human-specific inversion of chromosome 18. A further example of the impact of segmental duplications on karyotype and genome evolution in primates, Human Genetics. 115, 2004, 116–122.